Hey folks, I'm Ethan Polliner, co-founder of Cultra, the nutrient analytics platform for agronomists and growers. Today's video will focus on the role that arthropods play in a healthy soil environment. Let's dive into it. Arthropods are the most abundant group of organisms on Earth, representing roughly 3/4 of all living species. Their segmented bodies, jointed limbs, and chitin-rich exoskeletons allow them to thrive in nearly every environment. Because their exoskeletons do not stretch, arthropods must shed and replace them as they grow. Soil systems rely heavily on arthropods. Their life cycles, feeding habits, and movement patterns determine how organic matter is broken down, how nutrient cycling proceeds, and how air and water move through the soil profile. We'll begin with their basic biology, transition into their classification, and then explore ecological roles and the key groups that drive soil health. With these fundamentals in mind, we can first examine the basic biology and life cycle patterns that shape how arthropods function in soil systems. Most arthropods have 3 body segments, beginning with the head, following the chest, otherwise known as the thorax, and ending with the abdomen. Their growth depends on molting, which allows them to shed a rigid exoskeleton and produce a much larger one. Their typical life cycle progresses from egg to larva to adult, and many species spend all stages in or on the soil, while others rely on the soil for 1 or 2 of those stages. This life cycle structure helps determine how each group interacts with organic material and soil structure. Now that we understand their basic structure and life cycle, we can explore how soil-dwelling arthropods are grouped into major classes. Soil arthropods belong to several major classes, each with distinctive traits. The class Arachnida includes mites, spiders, scorpions, and ticks, many of which are predators or detritivores in our soil ecosystems. Chilopoda includes centipedes, which are fast-moving predators that help regulate other soil invertebrates. The class Diplopoda includes millipedes, which specialize in breaking down leaf litter and other coarse organic debris. And the class Insecta encompasses a wide range of species such as termites, dragonflies, cockroaches, lice, bees, wasps, and ants, many of which spend significant parts of their life cycles in the soil or litter layer. And lastly, the class Malacostra contains sow-bugs and pill-bugs, which are crustaceans adapted to moist soil habitats and play important roles in decomposition. Understanding these classes helps connect structure to function and sets the stage for exploring how arthropods collectively influence soil processes. Now that we know who is present, we can focus on what arthropods do and how their actions actually shape soil systems. Most soil arthropods, especially those in the litter layer, act as shredders that tear up organic matter into smaller fragments. This increases the surface area and allows bacteria and fungi to colonize and decompose plant debris much more effectively. Their feeding produces fecal pellets that are nutrient-rich and break down rapidly. And these pellets also become hotspots for microbial activity and accelerate nutrient mineralization. Arthropods also shape soil structure. Their fecal pellets, burrows, and secretions bind soil particles into aggregates. Stable aggregates improve aeration, infiltration, and resistance to erosion, and are core indicators of soil health. As arthropods move through the soil, they redistribute organic matter vertically, carrying litter material downward into mineral horizons where microbes can access and process it. Their feeding choices influence microbial communities as well. Many arthropods selectively graze certain fungi or bacteria, which can shift microbial dominance and alter decomposition rates. Predatory species also suppress soil pathogens by consuming pest nematodes or feeding on fungal spores associated with disease. Even when organic matter is abundant, some arthropods consume living roots, as seen with root maggots and cicadas. Others create burrows that influence soil moisture and gas exchange, forming microhabitants that protect microbes from drying or oxygen limitation. With these broad ecological functions in mind, let's examine the major arthropod groups that most strongly influence soil health. To begin with, let's focus on mites. 2 groups dominate soil mite communities. Orbitid mites inhabit litter layers, surfaces, and even plants such as mosses and lichens. They feed on fungi, algae, and decaying plant material, and some consume nematodes and dead springtails. Their numbers are extremely high because females reproduce without males. Adult orbitids have tough exoskeletons that protect them from most predators except ants, beetles, and reptiles. Their feeding and pellet production contribute significantly to nutrient mineralization and aggregate formation. The next type of mites are gamecid mites. In contrast, these are active predators. Many species in this group specialize in hunting nematodes, springtails, fly larvae, and other microarthropods. Because their population size depends directly on availability of prey, they are useful indicators of soil health. When gamecid populations are high, Soil food web supporting them is usually diverse and active. Their method of feeding, which involves injecting enzymes to liquefy prey tissues, helps regulate nematodes and other small organisms that influence plant and microbial dynamics. The next group are springtails. Springtails are extremely abundant, often reaching densities of up to 100 individuals per square inch of soil. Their forked appendage, called the furcula, allows them to spring backward when disturbed, sometimes traveling up to a yard. Species vary in eyesight and body structure depending on whether they inhabit surface litter or deeper soil layers. Springtails primarily feed on bacteria, fungi, and decaying organic matter, and are key contributors to nutrient cycling. They also serve as a major food source for predatory mites and help shape microbial communities through selective feeding. And the final group are termites and ants, who are among the most ecologically influential insects in soil environments. Ants have opaque bodies, functional eyes, and long legs, while termites have soft, translucent bodies, short legs, and no eyesight. Both groups are effective shredders of surface organic matter. Termites specialize in digesting cellulose, which opens up pathways for bacteria and fungi to further break down those plant residues. As they construct mounds and tunnels, both ants and termites mix large volumes of soil, sometimes up to 6 tons per colony each year. Their tunneling improves aeration and water infiltration and creates microhabitants that maintain moisture and temperature stability. This physical restructuring enhances root growth, microbial activity, and nutrient cycling. Together, these groups illustrate the wide range of contributions arthropods make to healthy, functioning soil systems. Arthropods are central to the soil food web. Their abundance, feeding behaviors, and movement patterns influence decomposition rates, nutrient mineralization, microbial community structure, aggregate stability, soil aeration, and water movement. High diversity and activity of soil arthropods, especially mites and springtails, often signal a biologically active and resilient soil system. Understanding their role helps us better interpret soil health and guides more effective soil management. I'm Ethan Polliner, the co-founder of Cultra, the nutrient analytics platform for agronomists and growers. If you're interested in learning more about soil biology and soil chemistry, visit our YouTube or LinkedIn for more informative videos. Thanks. We'll see you in the next one.